Here we can see normal values of the semen analysis that are being used as reference values. So in 1999 the WHO had given these values. They were little more, and now because fecundity and fertility status they are decreasing, so keeping it in consideration, WHO has reduced the values, and the values now they are like volume, it should be equal to 1.5 ml or it should be greater than 1.5 ml. Previously it was 2 ml. So similarly pH of the semen should be at least 7.2 or it should be greater than 7.2 any value. In 1999 criteria it was 7.2 to 8 only. So sperm concentration it is also reduced from 1999 criteria. In 1999 there were normal sperm concentration was 20 million sperm per ml, but now it is set to 15 million sperm per ml or greater than that. And then total sperm count per ejaculate, that is the total number of sperms in semen. So they are now equal to 39 or greater than 39 million per ejaculate, and previously it was 40 million or more than 40 million per ejaculate. Similarly sperm morphology it is also decreased, and previously the criteria was greater than or equal to 15% normal forms, but now it is only 4% sperm. If they are present in normal form, then the person would be considered as fertile. And then for vitality, that is the living or sperms. So for living sperms, the now criteria is greater to or equal to 58% sperm should be in living form. Previously it was 75% sperm they should be uh, living. So for white blood cells present in the semen, now the criteria is that they should be less than or equal to 1 million per ml. Previously it was same. And for motility it is total motility, now it is equal to or greater than 40%, including the rapid progressive motile and non-progressive. And previously it was greater than 50% or equal to 50%, and they were including rapid and slow moving sperms. And progressive motility it is 32% or greater than that, and previously it was for only rapidly moving sperms it was greater than 25 or equal to 25%. And then the status of the antibodies, presence of antibodies. Now the criteria is similar to the criteria of 1999. They should be less than 50% sperm. If they are adhered, then the person would be considered fertile or normal. And it is for MAR test, that is mixed anti-globulin reaction. And then immunobead test, the sperm they should be less than 50% sperm they should be bound with beads. So the antibodies, it means they should be binding to less than 50% sperms. So the criteria is same in 1999 and 2010. So we are following these criteria that are of 2010. That is the semen volume, it should be equal to or greater than 1.5 ml. pH should be equal to or greater than 7.2. Sperm concentration should be equal to or greater than 15 million sperm per ml, and the total sperm count per ejaculate or total number of sperms per ejaculate they should be equal to or greater than 39 million, and sperm morphology should be equal to or greater than 4% normal sperm. Vitality or living sperm they should be equal to or more than 58% live sperm, and then white blood cell they should be less than or equal to 1 million per ml. The total motility should be 40% or greater than that, and progressive motile sperm, they should be equal to or greater than 32% of the total. And the mixed antiglobulin reaction MAR test, the sperm should bind with less than 50% sperm should bind with the particles, and in immunobead test less than 50% sperm should bind with beads. That are the normal criteria. And the normal values of various biochemicals in semen that are determined by WHO, and they are same in 1999 and 2010. And for total fructose, it is a marker of seminal vesicle because it is released from seminal vesicle and being used for the energy purposes of the sperms. So they should be, fructose should be equal to or greater than 13 micromole per ejaculate. And total zinc, it is released from prostate, it should be greater than or equal to 2.4 micromole per ejaculate. And zinc is acting as the cofactor for various enzymes. And then total acid phosphatase, they are also released from prostate marker, prostate gland, so they act as prostate marker, and they should be are equal to 200 unit per ejaculate. And we know the acid phosphatase, it helps in liquefaction of the uh, semen, and if prostate is not releasing phosphatase in normal concentration, then the liquefaction cannot occur and the sperms they cannot get free out of the thick viscous semen. So these phosphatases, they help to degrade the uh, protein fiber present in the semen fluid in order to make it more fluid. And then total citric acid, it is released from prostate gland, and it should be greater than or equal to 52 micromole per ejaculate. Then it is seen that citric acid may be involved in direct or indirect metabolism of the spermatozoa in the or citric acid cycle. So the citric acid should be greater than 52 or equal to 52 micromole per ejaculate. And then alpha glucosidase, it is released from epididymis, and it is helping to uh, digest disaccharide to glucose, and it should be present equal to or greater than 20 micro unit per ejaculate. And then carnitine, it is also released from epididymis, and it should be present 0.8 to 2.9 micromole per ejaculate of the semen. Carnitine is actually a biologically active amino acid, and it helps the fatty acid to enter into mitochondria for producing energy by the process of oxidation. If these contents they become less in seminal plasma, it means these tissues they have, or these glands they have infections. There could be infection in prostate glands. If prostate markers are less, it could be infection in seminal vesicle. If markers of seminal vesicle are less, and could be any infection in the epididymis if epidermal contents are less. These contents they are assayed or estimated by using calorimetric methods or ELISA directly. So we have different terminologies that are used for defining the conditions of or status of semen. They are normospermia. If the volume of the semen is normal, it is equal to or greater than 1.5 ml. And aspermia mean if there is no semen volume, it means the person has not ejaculated anything. And then hypospermia mean semen volume is less than 1.5 ml. Hyperspermia if semen volume is greater than 6 ml. Azoospermia. Azoospermia is when there is no spermatozoa in semen, there is no sperm in semen, living or dead. And then oligospermia, it is when sperm concentration is less than 15 million per ml. And there could be polyzoospermia if there is concentration of semen is more than 200 million per ml. Then there could be asthenozoospermia if motility of the semen is less than sperms are less than 40%, that is the progressively motile and the non-progressively motile collectively if motility is less than 40%, or the progressively motile they are less than 32%. There could be a condition that is called as teratozoospermia if less than 4% of the sperms are normal in their morphology. They have normal head, normal tail, and normal flagella. There could be leucospermia if leukocytes are present more than 1 million per ml, and there could be leukocytes or WBCs. So if WBCs are present in semen more than 1 million per ml, then it is called as leucospermia. There could be hematospermia. Hematospermia is if red blood cells are present in semen in any quantity. And there could be necrozoospermia. There could be dead sperms, and if they are more, then it is called as the if the number of dead sperms are more than 42%, then it is called as necrozoospermia. There could be a condition that is oligoasthenoteratozoospermia. Oligo means if the number of the sperms are less than 1.5 million per ml, the motility of the sperms is less than 40%, and the uh, morphology of the sperms, normal sperm is the normal sperm percentage with reference to morphology, they are less than 4%, then this semen would be classified as oligoasthenoteratozoospermia. So these three conditions they could be present in single semen sample, that is number of sperms are less than reference value, motility is less than reference value, and the morphology is also less than normal values or reference values. So they are called as oligoasthenoteratozoospermia. We know azoospermia, that is no semen present in the no sperm present in semen, could be due to the blockage of the reproductive tubes, vas deferens, or any other tube, and there could be another type of azoospermia in which the um testicular they fail to produce these sperms. So there could be Sertoli cell only syndrome when the um, uh, spermatogonia cell they could not be migrated from the yolk sac into the gonads during development. So these conditions they could be classified into testicular failure, or they could be classified into the blockage of the uh, vas deferens. Similarly, oligospermia could be mild, that is, if sperm concentration is uh, more than 8 million per ml, and could be severe if the sperm concentration is less than 8 million per ml.